Hello, welcome back to our last section of chapter 4, talking about classification. So this chapter is just kind of reviewing hopefully some prior knowledge from your general biology class of how living organisms are classified, and then what are some techniques that we use to currently classify bacteria based on different kinds of characteristics. So first, before we jump into kind of tracking living organisms, just remember we have three, or taxa are groups of living things that have similar characteristics. Taxonomy is a study of those living things, or studying the classifications of those living things. And if you remember, there are all of these hierarchies, these different taxa. So domains, kingdoms, phylum, order, family, genus, species. So you should know those. I share the mnemonic of dumb kings play cards on fine green sofas. You can use that if you want to, or if you've learned another one, but you should know that sequence, that domains, right? We have eukarya, archaea, and bacteria are the three domains of living things. And then each of those domains have their own kingdoms, and each kingdom has their own phylum, and each phylum has their own class, and so forth and so on. So on the next couple slides, we're going to track what's called a vector, which is something that can pass on disease. Itself is not a microbe, but just to give you a feel for how a single organism can be classified into these different groups based on their characteristics. This isn't something I'm going to need you to be able to do, but I just want you to be familiar and comfortable with this is how it works. All right. So first, we are going to start off with our domains, right? So we have our three domains, bacteria, archaea, and eukarya. And this particular organism is going to fit in the eukarya domain. That tells you that it has a nucleus, membrane-bound nucleus, and membrane-bound organelles. Okay? So then of the kingdom or the domain eukarya, there are three kingdoms within the domain. So there are things that that are made up of eukaryotic cells that have different characteristics, right? We have animals, we have plants, and we have fungi. So this particular critter we're going to be tracking is an animal, which means it's heterotrophic, it's multicellular, it usually has locomotion, not always, and some other characteristics of animals. Plants are multicellular, photosynthetic, usually sessile, which means they don't move around, they don't have locomotion. Fungi are single or multicellular, they're heterotrophic in that they do not make their own food, and they have some other characteristics like a chitin cell wall and things like that. Sorry, I had to sneeze, I paused. Back, hopefully. Close the door so the allergens wouldn't come in. All right, so this particular critter, it's a eukaryote, which means it has a nucleus. It's an animal, which means it's heterotrophic in all the characteristics the animals have. And then of the kingdom Animalia, there are phyla, there's different groupings of animals. There's vertebrates, arthropods, like jointed-legged animals, flatworms, and roundworms. So if we're to have our specimen, we would look at it and say, okay, it doesn't have a vertebrae, so it doesn't fit here. It's not a flatworm and it's not a roundworm. It's got some jointed appendages. Okay, so it's going to be in the phylum Arthropoda. Arthropods means jointed legs. All right, so I'm still looking at my little specimen here. You don't know what it is, but in my brain I can picture it. So of the arthropods, now we're going to zoom in a little bit closer and say, okay, so of all the things with jointed legs, is it an insect, which means it has 2 body parts and 6 legs and some other things. I'm not super strong with my insect classification, but it's not insects. Crustacea has like 12 body segments, multiple legs, different kinds of things. It doesn't have those. Arachnids, 3 body segments, 8 legs. Yeah, okay, so it checks those boxes. So it's going to be in the class of Arachnida. And when I go to the next slide, you're going to see what critter we're talking about. So of Arachnida, of all of the arachnids that fit that category, we can further break it down into the orders. We have different types of arachnids. We have ones that are like scorpions, we have ones that are like mites, we have ones that are like ticks, and we have ones that are like true spiders. And this particular thing we're going to look at is a tick. It's not a scorpion, it's not a mite, and it's not a true spider. So based on its physical and biological characteristics, it fits into this order of parasitiforms. All right, So now let's zoom in even further, see if we can make some smaller groups. So the next category or taxa was a family. So of this particular group, we have two kinds. We have hard ticks and we have soft ticks. Now, at this point, you're probably going to have to be an entomologist to start discerning the differences, because I would probably be stuck on, well, I might be able to do order. If I had maybe a dissecting scope, I could figure stuff out. I wouldn't know the difference between a hard tick and a soft tick without learning about it or doing more research. So this is where we start to get into the specialties and people who really study these for a living. So apparently our deer tick that we're going to eventually get to is part of the hard tick family. All right, so now you're an entomologist and you're like, okay, so of hard ticks, there's a couple different types of hard ticks. There's this group, a genus, there's this group, and there's this group. So apparently our deer tick fits more into the category of this Ixodes genus in the Ixodidae family in the Parasitiformes order, right? So we can go all the way back up. So then of the Ixodes genus, there were three groupings that are different enough that they get their own category of species. And this particular one that we are trying to find and categorize is called Ixodes domini, which is a deer tick, which is the vector for Lyme disease. So there's a little bacterium that lives in their saliva, and if it bites you and mixes with your blood, the saliva with the bacterium can get in there and give you Lyme disease. And the other two, if we were to have the knowledge to be able to identify enough, we would say, no, this particular specimen I'm looking at does not match Ixodes pacificus or Ixodes ricinus. It matches Ixodes domini. We found a deer tick. Yay! So that is our classification. So domain, kingdom, phylum, class, order, family, genus, species. Okay, so when we start, once we figure out kind of what the thing is, all the way down to its last two groupings, the genus and the species, that is what we use when we name things. So when we do scientific naming, scientific names in accepted naming conventions have two names. It's called binomial nomenclature. So binomial two-name nomenclature naming system. So a two-name naming system. It includes the first name is the genus that it belongs to, and the second name is the species that differentiates all of the other organisms within that genus. So I have some rules for scientific naming. The genus name is always capitalized. The species name is not capitalized. And the name, when writing it, is either underlined or italicized. So that helps to kind of pop that name out of the typical text of whatever it is that you are reading. And then lastly, once you've used the full name, the genus and species written completely out in your description, you can then abbreviate it by abbreviating the genus, just the first letter of the genus, but then spell out the second, the name of the species. And so let's do some examples. Okay, so we've all heard of E. coli before, usually, hopefully, we've all heard of E. coli. So its full name is Escherichia coli. So I have capitalized the Escherichia because that's its genus, lowercase to the coli because that's its species name. If I were to write a paper talking about E. coli, I would spell out, say in the first paragraph, Escherichia coli, and then every time after that in my paper, I could abbreviate it E. coli. And its name is derived from Theodor Escherich, who was the discoverer of it, and that it lives in the colon. So scientific names can be a combination of kind of paying homage to the person who discovered it, or descriptive terms about where it lives or what it does, or maybe a shape or something like that. People are trying to get away from more people names and trying to stick to more descriptive terms. But once you discover something, you have the right to name it whatever you want. Here's another example that I've done in italics. So Streptococcus pyogenes. So it's in the genus Streptococcus. It's the species Pyogenes. So once you spell it out completely, then you can abbreviate S. pyogenes. The name indicates more descriptive characteristics. So strepto means chain. Coccus means circles, like sphere-shaped cells. So Streptococcus might look something like this, a whole bunch of bacteria in a chain. And then pyogenes is a root word that means pus. So if you get an infection with S. pyogenes, typically the wound is going to be gross and pussy. Ugh, yeah. So those are examples of scientific names and how to properly use them. So when you are writing your case studies and your answers in your case studies, to get good practice, why don't you try using these naming rules? Proper capitalization, italics, underlined, and abbreviation when necessary. All right, and then here's just kind of a cool picture showing the domains. I had mentioned earlier that the branching of our tree of life is that our common ancestor branched into bacteria and this branch pretty early on, and then archaea and eukarya branched off of that branch because they're finding more similarities between archaea and eukarya than they are archaea and bacteria, which is weird because both of these are prokaryotic cells lacking a membrane-bound nucleus. Okay, the rest of the slides are just going over different ways that microbiologists have to help with classification and identification of organisms. One of them is called Berge's Manual. So there's a Berge's Manual of determinative bacteriology and systematic bacteriology. We actually have this set of Berge's manuals in our microbiology lab on campus. And so when students have questions about a particular organism, they can look up all of the knowledge that has been gained in the whole world of microbiology is published in these huge volumes. Their books are like this thick, and there's like 5 of them in the set of the Berge's manuals. So it's all the knowledge that has been gained and approved and coordinated by microbiologists all over the world. You can take a look at physical characteristics. So organisms based on the shape of the cell. So for example, these guys are what we call bacilli or bacillus. So if they are rod-shaped, we can say, oh, okay, so maybe all rod-shaped bacteria are similarly connected to each other. These guys are called cocci, which are the spherical shaped cells. And then these ones are called spirillum. We'll see these terms in chapter 11 when we classify them, learn shapes and things. So we can classify organisms based on their physical characteristics at the cellular level. This is called cell morphology. We can also classify them based on how they look when they're growing on a solid media. This is a petri dish with a whole bunch of different kind of bacteria, and this is what we call colony morphology. What do things look like once they're growing on the surface of a nutrient agar? And so we can see they circled like the number 3s here. All of the number 3s kind of have a circular colony. They almost have like an outer ring with a darker center. So that tells you that the number 3 bacteria is different than the number 4 bacteria, which is a lot bigger of a colony. It's a solid white, and it doesn't have a little ring outside of it, which is different than number 9 because it's yellow, which is different than number 1 because it's really big and kind of not quite circular. So these physical characteristics on the larger scale, this visual with your naked eye scale, looking at the growth of the bacteria on your petri dishes lets you know that you have more than one organism. So this is a really good thing to look for. So in lab, when you're growing things on your agar dish and you're hoping that it's only either E. coli or Staphylococcus epidermidis or the yeast, if you have something on there that doesn't look like what you've normally been seeing, it's probably a contaminant. And so you want to get rid of any, don't mess with the contaminants because they could mess up your experiments. So it's really good to start getting familiar with what these organisms might look like growing on the solid surface of your petri dishes. You'll be doing some biochemical tests. So sometimes we can classify and group organisms based on the results of how they do different metabolic pathways. So if your organism produces acid with gas when it's fermenting a particular sugar, that's going to be different than somebody that doesn't produce acid or gas as a byproduct. Or this particular one, this organism produces hydrogen sulfide and this one doesn't. So that could group them into different categories. You could use serological testing, so the ability for antibodies to recognize those surface proteins that could be on the outside of a cell wall, say showing no clumping here or clumping here, lets you know if you know what your antibody recognizes and you get clumping, then that's a good clue that you have what it is that you're looking for. Phage typing is specifically for bacteriophages, for viruses that kill bacteria. So here you plate out your bacteria that have the viruses in them, and all these little plaques, these kind of holes in the growth on the surface of the plate, they represent where all the bacteria have been killed by that particular virus. Viruses are very species specific. So if you have death to your bacteria with a known virus, that lets you know that you have that bacterial target that that virus can infect and kill. And then lastly, we have analysis, or not two more, analysis of nucleic acids. And this is probably the most modern form of classification outside of the physical characteristics and the biochemical pathways. They all help to support, but really comparing DNA and RNA really helps to say you are related to this guy over here because you share a large percentage of the sequences of your RNA and your DNA. And then lastly, you can use taxonomic keys, which are kind of just a flow chart to help you identify based on some of your physical characteristics, your metabolic pathways, maybe even some DNA characteristics, which isn't shown here, but it basically helps you rule out what your organism isn't based on the characteristics that your organism has, right? Okay, so that wraps up our chapter, the last part of chapter 4, because remember 4 was kind of our weird chapter. We started with measurement of microscopes, then we went on to staining, and then we finished with taxonomy, or yeah, classification. So, all right, we will move on to, we're covering a little bit of chapter 11 this week along with chapter 4, since it kind of goes well with this last piece of classification. So we'll learn a little bit more about different groups of bacteria. All right, I will see you then. Bye.